Welcome to the pod. We've got a fantastic field diary episode for you today. That's right, Cassidy Beach, Science Solutions Santa Cruz producer, guest hosts, and she is joined by Steve Haddock, senior scientist at the Monterey Bay Aquarium Research Institute, also known as MBARI. And today, you are going to get a front row seat to the deployment of a massive remotely operated vehicle aboard the RV Rachel Carson, which is one of MBARI's research vessels. So Cassidy and Steven and their team, they go out to sea, they collect footage, they collect audio. You're actually gonna see them pull some organisms up from the surface. You're gonna learn about the Monterey Canyon and its role in the global ecosystem. This is a really special, really adventurous episode of Science Solutions Santa Cruz. I'm Jonathan Hicken. Enjoy.
Hello again, Science Solutions Santa Cruz listeners and viewers. I'm Cassidy Beach, and we also have Steve Haddock here with us today. He's a senior scientist and marine biologist at the Monterey Bay Aquarium Research Institute. He's also an adjunct professor at UC Santa Cruz. Today, we're going to be talking about deep sea research, what that looks like, and we're actually going to go out on one of the boats. So stay tuned for that. Without further ado, hello, Steve. How are you today?
Hey, Cassidy. I'm doing great. How are you?
Great to have you here. I first just wanted to begin the conversation by asking, because I feel like there's two camps of people. Mm-hmm. There's people that look at the deep sea and they're fascinated by it. And then I think that there's a whole nother camp of people that often look at it as like this deep, scary abyss. So what initially drew you to study the deep sea?
I've been scuba diving since I was like 13 years old.
Oh, wow.
And I just was really into marine biology as a kid growing up in Southern California, going to the beaches down there. I didn't really think of it as like a potential career, but then in college, I had a really influential biology professor who encouraged me to check out graduate school. He basically sent me off to UC Santa Barbara, where there was a guy who studied bioluminescence. There was somebody who specialized in deep sea and somebody who specialized in like open ocean scuba diving. And so those three professors ended up being my committee and giving me a bunch of opportunities to study these rarely seen ecosystems, I would say.
And then where did your career take you after grad school?
So I was in grad school. Afterwards, I got a postdoc. It's called a postdoc when you finish your PhD. You spend a couple of years at the Monterey Bay Aquarium Research Institute. So I actually started there as a postdoc with another researcher, and then eventually got hired as a PI myself, as a principal investigator. So now
You've been
have
there
my
ever
own
since.
lab, and I've been there ever since.
Wow. Okay. And then why is the deep sea something that people should pay attention to? Why is it important?
I mean, there's a lot of reasons for me. One of the things is that it's by far the largest habitat on the planet. You know, you think of the ocean as like 2/3 of the planet is the ocean. That's really just the surface area. But the average depth of the ocean is 4,000 meters. And so that volume of living space is way bigger than all the land habitat added up. And so I think it's important to understand that it's not just a lifeless void down there. And the animals they're not a threat to us. They're actually they're connected to us in that the things that that people eat on their dinner plates are linked to deep sea animals through the food web. And also there's a lot of talk about carbon cycling that takes place in the deep ocean as a reservoir for carbon. So there's a bunch of examples. For me, I'm motivated just because of the number of unknowns down there. There's just so many species to discover and interactions to discover and really cool ways of making a living in what to us would be completely inhospitable, inaccessible habitat. But these animals, that's their happy place.
Yeah, I remember reading, is it true that 80% of the seafloor has not been like mapped or discovered yet? Is that true?
I mean, there are papers like that. I think, to me, a lot of the focus on the deep sea is the seafloor.
Mm-hmm.
And that's actually, if you just go straight to the seafloor, you're ignoring the actual deep sea. The deep sea is the water that's above the seafloor. So for me, yeah, mapping the seafloor is important, but to me, it's a lot more important to look at the animals that live between the surface and the seafloor. And there, the number is much, much larger than 80%, I think, of the space that we haven't explored.
And then you mentioned that they're important to us and what we put on our plates. Can you explain that a little bit more?
I mean, just a simple example would be a tuna or a swordfish that somebody might, care about commercially to go fish those. Those can feed on squid that live down in the deep sea, and they migrate up and down during the day. And then the squid in turn are eating other deep sea animals like lanternfish. You can't just skim off that surface of the ocean and say, we care about these animals, although we don't care about anything below that, because they're all interconnected and linked through vertical migration, but also just through the broad distribution of some organisms.
Right. Now, we actually worked together in the past because you had recently released a new book with one of your colleagues, and it was about light in the deep sea. Can you talk a little bit about that? Why do deep sea creatures create their own light? And what are the different kinds?
We wanted to show kind of pictures of marine animals that people may have never seen before, but we organized it in a way that was kind of how they interact with light in the ocean. So one chapter is about bioluminescence, about how they make their own light. But then there's 4 other chapters that are about transparency or iridescence and fluorescence and pigmentation, other ways that organisms alter, and sometimes we say like hack their environment, right? They're manipulating the light field so that it either gives them an advantage, makes them invisible, makes them hide. In the case of bioluminescence, so many of the animals in the ocean are luminous. We did a study using our submersibles where we have like 30 years of dive records where we annotate, catalog everything that we see on the dive down to 4,000 meters. And
You're in the dive gear going down 4,000?
No. Sorry.
Like, what?
This is a remotely operated submersible.
Okay.
I mean, I have been down to 1,000 meters in a sub.
Wow.
But for what we do here in the Monterey Bay it's called an ROV, a remotely operated vehicle. So as you will see, we control it from the ship with like a joystick. It's kind of like a video game. You're watching a screen, seeing what the sub is seeing and driving it around and looking at and for animals and then collecting them.
Okay.
So we took all those annotations. It's like almost 2 million animals that have been cataloged in that database. And then we mapped onto that which animals are bioluminescent or not. And we found that 3/4 of the things that we see from our sub are bioluminescent. And that ratio is the same all the way from the surface down to the deep sea. So it's really prominent. It's really widespread in the ocean. So it's for sure important. And what's fun is that there's a lot of different functions that things use it for. So anglerfish are kind of one that people might be familiar with. They have a little lure out.
Yeah, from Nemo.
Like moths coming to the street lamp, and but that's actually pretty rare in the ocean for that to be the function. A lot of them are defensive. So you shoot out a cloud of sparkling bioluminescent material while you swim away, and your predator is like confused or attracted to that smoke screen. A bunch of different kinds of animals do that. Some of them just flash. We think it might be like the bright coloration on noxious animals like monarch butterflies. They have bright orange color, which seems like it wouldn't be a good idea. But if you also don't taste good, then that advertises. Like there's invisibility cloaks that animals use to hide their silhouettes, hide their shadows from things that are looking from below. And what's cool is that some animals have 3 like different functions. You can just see like they have different photophores, different light emitting organs on their body in different places. And so one animal can be using bioluminescence in 3 different ways.
Wow. If bioluminescence is that abundant in the ocean, can we see it here in Santa Cruz?
Yeah.
Could I go to the beach and see it?
Almost any night, pretty much any night, there will be like some sparkles in the water that you'll see.
Really?
You want to go down like on a moonless night. And then every once in a while, there are these big events. It's kind of like a red tide, but it's with bioluminescent species of single-celled microscopic algae. And then like the waves will be making blue flashes when they crash on the beach, or when you walk on the sand, your footsteps will be leaving little prints of light. It's more rare, but actually here, like especially off Santa Cruz, there's what's called an upwelling shadow, where the water is kind of calm right up in that nook of the bay up there.
Mm-hmm.
And so these single-celled algae have a chance to reproduce and grow and kind of multiply in that little incubator up there. And so you can get really high abundances just right in Santa Cruz.
I've only experienced bioluminescence once. I was on a night dive, and we all kneeled on the bottom of the ocean, and we started drawing in the sand. But
Wow.
I've never seen it in any other circumstance, so yeah, I'll have to go check it out. Why is Monterey Bay Canyon such a good place to study the deep sea and all these other things that you're discovering?
I mean, I work on a lot of different sort of aspects of deep sea biology. So genomics, bioluminescence chemistry, but also behavior and functions, and diversity, like cataloging species diversity. And so for a lot of that, we can use technological advances in other fields to amplify our ability to study it. So low light cameras, like we have really good imaging capability now with like high res color, low light cameras. And then there's like the genomics revolution, where we can sequence entire genome in like practically in an afternoon now. And there's also even AI tools are really helping us annotate those millions of images that we see. So classifying the organisms. So we're applying a lot of technology to it, but it doesn't replace actually going out into the deep sea and finding the animals and like looking at them in their own environment. You can't just sit at your computer and make like a discovery about life in the deep sea as much as you can if you go out, with a ship and a submarine. So access to the deep sea, though, is not so easy. If you live in Florida, the shelf, it's just like gets gradually deeper.
Mm-hmm.
So you have to literally go on a ship and then go overnight, like 24 hours offshore to get into deep water. And off here, we can go in about an hour and a half and
Wow.
in deep water, because the canyon brings the deep water up to shore.
What do you classify as
So-
deep water?
I mean, typically it sort of starts at 200 meters, but we can get to 1,600 meters, so a mile deep is where our station is that we're going to go to on the ship. And our institute is located right in the middle of the canyon, of the bay, which is where the canyon comes up to shore. If you were to drain all the water out of the bay and stand on the edge of it and look down into the canyon, it would look just like you're looking at the Grand Canyon. It's the same scale
Really?
as the Grand Canyon. And so that's making deep sea organisms really accessible to us to study. So we can go out in the morning, collect things, look at things, be back at shore by 4:00 in the afternoon, and still work on the animals. And that access to the deep sea is really important and really special about this area.
And then when you're bringing these creatures up from how deep again?
As much as 4,000 meters.
Yeah,
almost.
4,000 meters. Are they surviving? Because there's so much difference in pressure there.
Yeah.
And then once you get them on board, what are you doing with them?
We actually have an NSF project to study the effects of pressure on these jellies, because they don't really have any reinforcement that you can see. They're just so diaphanous and fragile. If you opened a scuba tank at that depth, the water would go into the tank. The air would not come out of the tank. It's like more pressure than the inside of a compressed air scuba bottle.
Mm.
And so their membranes, just if we took like our cell membranes down there, they would just become brittle and crack. They wouldn't be able to function because of the cold temperature. It's colder than your refrigerator. It's barely above freezing, and it's 400 times atmospheric pressure
Wow.
down there. So we have a project where we were looking at the chemical composition of their membranes and how they alter that so that they work down there. And the trade-off is that if you bring them up to the surface, their membranes can start to melt. And so,
Melt?
yeah, they
'Cause
just
it's so
kind of
used to the
fall
cold.
apart. So yeah, if you think of a stick of butter, like if that's soft, so you could spread it on a piece of toast, that's like functional cell membrane. Put it in the fridge, suddenly it's brittle and cracks, and it's not bendable. Or if you set it out in the sunlight, melts and just turns into liquid. It's kind of the same thing. You have to have this just right condition where it's like soft spreadable butter
Okay.
for the membranes to function. Otherwise, they can't handle too warm a temperatures, too low pressures, or too high pressure. Having said that, that is for the things I would say that are below about 2,000 meters. We can actually bring the 1,000-meter deep organisms up, and the pressure doesn't seem to be that much of a factor. So as long as we keep the temperature cold, they seem to do okay. And in fact, the aquarium, the Monterey Bay Aquarium, which is like our sister institution down in Monterey, they have on exhibit some of these really fragile deep sea animals that I never thought they would be able to do that.
That exhibit is incredible.
Yeah.
Yeah.
It's really one of a kind. And they just collected, like 2 days ago, they just brought back this really rare deep sea ctenophore, and they had it on exhibit. I don't know if it's still there, but
Can you describe what a ctenophore is?
sorry, yeah. Dropping my jargon. Ctenophores it's one of the main groups of animals that I study.
Okay.
They're also called comb jellies because they swim with these little cilia, these paddles that are in rows along the side of their body. And the aquarium is one of the few places that you can actually see them alive. They're really abundant in the ocean, but they're also not familiar to most people because they're so fragile. So if they wash up on the beach, sometimes there's a species that's called sea gooseberries. It just looks like a little clear grape on the beach. And then again in Woods Hole, like on the East Coast, they have this one called the sea walnut that they call it, but it's more fragile but larger. But you can see if you're swimming in the water. We did a study where it's the most remote animal to the rest of the animals. It's like the most distant relative, the sister to all other animals, including sponges. Like we're closer related to a regular jellyfish than we are to these comb jellies. But it's just a really interesting group of animals that's in the ocean. But it's hard to study because they're so fragile.
What animal are we mostly closely related to in the ocean?
Well, besides fish, like invertebrates,
Right.
there's these things called salps, which are in their chordates. They look like a little transparent barrel. They pump water through their body and filter out food
Okay.
from the water.
Are these also deep sea creatures?
They can be, but they reproduce by cloning, so they can get to be large numbers and high abundances. And they look kind of like an ammo belt,
Okay.
like the old thing with like,
Yeah.
so they make these long ribbons of tubes connected to each other, but it can be like 30 feet long, or it can look like a big cinnamon roll if it's curled around. There's some that are helical, so they're spirally.
And we're most related to these things?
We're most related to those
Okay.
things.
Interesting.
I mean, of the invertebrates, they're called urochordates because they have like a primitive cord, backbone. They're like the next group out from the vertebrates.
If you haven't visited the Seymour Marine Discovery Center at UC Santa Cruz in a while, or ever, you really should come visit us. We are right on the bluffs of the beautiful Monterey Bay, with an aquarium full of local species and a bunch of hands-on science exhibits that help you understand what's happening in our ocean and along our coast. We are connecting the incredible research happening at UC Santa Cruz with real-world solutions for our coastal community. We don't want science to just sit on a shelf. We want it to shape decisions, inspire kids, and help our community feel equipped to act. So if you care about the future of our ocean and our coastal community, or you just want to experience a place that makes you feel a little more connected to it, come visit us at the Seymour Marine Discovery Center. Science Solutions, Santa Cruz.
Well, I feel like you've seen a lot of alien-looking things. What is the coolest?
Don't ask me my favorite. You're gonna ask me.
Not your favorite, but like the most bizarre thing, something that you saw and you were like, "What is that?"
We just described a species of swimming snail that we called it for a long time the mystery mollusk, and I wanted to call it something about Dr. Seuss because it looked like a Dr. Seuss kind of creature. It has a big hood in the front that it uses to catch its prey like a Venus flytrap. But then it's almost like a little horse-like body, and then these flappy fingers sticking off as a tail, this fleshy tail.
Okay.
And it's bioluminescent. So when we filmed it with our low-light camera, like its entire surface of its body just glowed with kind of like, looked like stars. But yeah, when we first saw that, we were like, is that a sea cucumber? We didn't even know what like group of organisms it was in. But then as we studied it, we found out it's like, again, it's like a really distant relative of other nudibranchs. Like, I don't know if people have seen these really colorful sea slugs.
Mm-hmm.
It's a very distant relative of those guys. It's its own branch. And instead of crawling along the bottom, it swims in the water column. So that's one of the more bizarre ones. I would probably, for most people, the siphonophores are the weirdest. So these are these long tubular jellyfish relatives that, again, can be like 100 feet long, but just the size of like a broom handle.
Wait, are these different than the ones that were our closest relative, or?
Yeah, different.
Okay, totally different.
Another thing, they have all their swimming bells at one end, so they're like these little pulsing things that look almost like a chain of jellyfish. Those parts can't feed at all, though. So the rest of it that looks like a tail has a bunch of stomachs hanging down, and each stomach has its own tentacle hanging down.
What in the world?
And so they're connected by a tube, so a stomach will catch its prey, digest it, send the nutrients up to the swimming bells that are at the other end. So those are pretty weird for a lot of people, and we've named a bunch of new species. We just submitted a paper with 2 more species descriptions.
I wanted to ask about that.
Guys.
Yeah, how often are you guys actually finding new species? And then, like, what is the naming process? Do the people who actually saw it first get to name it, or how does that work?
It's kind of whoever does the legwork to get the paper ready, and usually we'll include whoever saw it first, but not necessarily always. Every dive will see something that doesn't have a scientific name yet.
Really?
But whether it's new to, like, we've seen them before. They're kind of like our old friends, but they still haven't been formally described.
Okay.
So we'll see some for sure when we go out on the ship.
Oh,
I'll
cool.
show you some of the ones that are not named yet, or some of the ones that we recently named. We should see a bunch of things that we just have named relatively recently.
So you guys kind of do them in batches? Like, we found all these things and we're naming all of these now?
Like, if you found some new species of crows, you would describe, like, here's, summary of the known species of crows, and here's 2 new species that we found. So you put the crow paper, and then you have the hummingbird paper. And so here's like the known species of hummingbirds, and here's a couple new ones that we found. So you try to kind of batch them like that. But the hard part really is doing the historical literature search and making sure nobody's seen this before. What's its closest, relative? What group does it belong in? So we'll always do, it's called the morphology, right? That's describing how physically the shape of all of its structures of its body, but then also the genetics. So we'll sequence a bunch of different genes from it and put it into a tree to place it in context, like, oh, it's a close relative of this other group, or, it's in its own branch of the tree. And that lets you put it into a family or one of these higher level classification things. And then you try to think of a tricky name to put on it. And we have some new instruments that are going out on autonomous vehicles that have like microscopes on them. So they'll go out and they'll photograph all of these little, like the bioluminescent little tiny things, copepods. In one deployment, they're autonomous, so we can program them from shore. They come up and you can send it a new mission and see where it is on the map and see what its depth profile was.
Wow.
But one deployment of these things, it can come back with like 2 million things that it saw.
And these are all like 2 million individual photos?
Yeah. Well,
Okay.
It actually is pretty smart. So like if there's 2 things in the frame, like if you took a picture out the window and there's a bird here and, a snake or lizard over here, it would just save those little boxes. And so it just saves, they're called the ROIs, the regions of interest. It doesn't store the whole image, every image. It just stores the ROIs from them. But obviously a person can't go through 1 million images, and say, yeah, that's a copepod, that's a fish, that's a whatever. So we're using machine learning to train models and then classify all these data sets automatically. So we do have a big effort for a bunch of different projects. We have a drone project where we fly surveys over the water.
Okay.
And these tools can classify that there's a seagull, there's a seal, there's a great white shark, whatever it is that we see from the drones. So we're using a lot of AI to help streamline that process, so one person doesn't have to go through and try to tag everything.
So how often are you guys going on these research expeditions? Is the main goal to find new species, or like does each one have a different goal? How does this work?
It's averages about once a month. We have day trips where, like I was saying before, we can go out in the morning, do a dive down to 1,000 meters, come back in the afternoon, work on the animals. But then we have longer trips, which we're going to go on in a couple of weeks, where we go out for 10 days or something, or 8 days, and we can kind of leapfrog our way all the way offshore, 160 miles offshore, or down to San Diego, and do this big loop where we're diving in each of those places along the route. And those I have maybe like twice a year. I'll have one of those longer trips. And then we also charter a dive boat in Hawaii so that we can go out and collect shallow samples. What we do with them, we have a lot of different projects in the lab. So one of them was that project with the lipids that I was talking about, the membranes adapting to the deep sea.
Mm-hmm.
For comparison, it's good to have samples from Hawaii, like shallow warm water, to compare to cold deep water. And then we even have cold shallow water, like in the Arctic and the polar regions. And so you can look at the properties of those membranes and the genes that are involved, and say, like, this property is correlated with pressure. This property is correlated with temperature. So one of our projects, I have a grad student through UC Santa Cruz, and I had a former grad student who just finished, who was looking at the lipid end of it. Then we're also looking at the bioluminescence chemistry.
So there's a lot going on board.
Genomics, population genetics. Yeah, we have a bunch of different projects. And so for each project, we'll have like, we need to get that species, and we need five of these guys to kind of fill out that comparative matrix of things from different environments. One thing I would say is that, like, the technological advances have really made it so we don't have to, like, when people were first studying bioluminescence, for example, they would have to collect like 100,000 jellyfish and grind them up and try to purify the chemicals out of that just to characterize them. Now we can get one jellyfish, we can get one thing, we can sequence its genome and basically find out almost everything we need to know from one specially targeted specimen. So it's a really low footprint for research. People are always like, do you let them go after you collect them? It's like, not really. We're immortalizing them.
But at least we're not grinding them up.
Yeah, at least we're not mowing thousands and thousands of them. So I like to think of it as we're making them immortal by freezing them and sequencing their genome. So they basically contributed the information for their species to our knowledge.
And why is it important to understand these creatures in the deep sea?
I mean, I think part of it gets back to the fact that it's the largest habitat on Earth. Most people think of it as lifeless or only has ugly, worms that are just like burrowed into the seafloor. But there's this whole really dazzling array of biodiversity that lives down there that most people don't know about. And I think showing them more and more of these animals and having them say, wow, instead of yuck, is part of the journey towards having people conserve the ocean and protect it. The more people are aware of things, then the more they will actually care about them. And so when you have people proposing, we're just going to mine the deep sea, or we're just going to dump, carbon dioxide down there to get rid of it, or pollution.
Understanding what's at stake down there.
Yeah, it's not just like throw it in there and forget about it, and it's not going to bother anything.
Well, I'm excited to get on the boat and see this all for myself. So what do you say we go down to Moss Landing and we get on the boat? All right, so we're now in the Monterey Bay on the Rachel Carson, and Steve's going to tell us a little bit about what we're out here today for.
Yeah, so we're going to do an ROV dive. This is called a remotely operated vehicle. This is the ROV Ventana, which means window. It's our window into the ocean. And there's a tether that connects this back to the ship, provides power from the ship to the vehicle. And so we're going to be sitting in the control room up in the bow, controlling this remotely as it's deployed. And we're going to go down to about 1,000 meters or 3,300 feet. The main camera is a 4K color camera, reconfigured to be mounted in this housing, and has this optical dome for the lenses. And then when we see stuff, we can either film it if it's doing an interesting behavior, if we see an interesting like interaction between different animals. But then we can also collect stuff for our genetics work and for our bioluminescence work. And we have two main ways of collecting. One is with these samplers here. It's a cylinder that you drive the vehicle all the way around the animal, close those two lids on top,
Okay.
top and bottom, and it seals it in there with that O-ring. And they can catch animals with their surrounding water, so they're very, very undisturbed. The other way is this nozzle where there's a clear nozzle that runs to a tube back into a carousel with 12 sample buckets in it. And that can rotate around, so you can put a different bucket under the nozzle, fill that one up, move it on to the next one, suck something into it. So those are our two main ways of collecting things. Great. Whoa.
Cute.
Such a cool texture. Yeah, it glows. It's really beautiful. All right, I guess let's suck it up. Get in there. Okay, there it goes. All right.
There's enough tissue for everybody on that. Everybody gets some of this.
There we go.
Yeah.
That was very good. That's fine.
So how many species did we collect in total today?
Probably got like 14 things, of which 3 don't have a name yet. 3 are undescribed. Yeah.
Okay. Do you guys take these species into this wet lab over here and then do all your work here, or are you guys mostly taking them back to onshore?
We unload the samplers in there and we transfer them to smaller containers, like a cooler, and then we bring them back to shore.
Okay.
Look at the keels. Oh, he's big.
It's another undescribed species.
So cool.
You have one of the coolest jobs ever.
I like it.
Okay, we are back in the studio now. That was amazing. Now that we understand what's down there, what's at stake, I wanted to kind of move into some of the threats that the deep sea is facing.
Mm-hmm.
I know that there's a push for deep sea mining, and to my understanding, that doesn't just impact the seafloor, it impacts a lot of other ecosystems. So can you talk about some of the threats?
It's kind of a hard time to talk about the threats because there's like all the logic in the world. It doesn't seem to percolate up into the policy domain so much.
It's a good way to put it. Yeah.
Like for a while, specifically about deep sea mining, I was a little bit encouraged. The International Seabed Authority is the UN, the United Nations sponsored agency that is supposed to oversee mining in areas that are not part of any country's boundary. So the middle of the ocean is supposed to be the heritage of all mankind. And so this ISA was the body that was supposed to govern that region. And the former president, if I can like be blunt, was pretty corrupt. He was like in cahoots with the mining industries that he was supposed to be regulating, but they just had an election and they got rid of him. And so they have this other person in there. And so for a while I was very hopeful about that. But now there is talk of having the U.S. permit mining in these open ocean regions, which are not our territory. And to fast track it also. And so to me, it's just really worrisome again that, no amount of knowledge or facts about the deep ocean and the potential risks and the potential consequences seem to move the needle, because it's, there's a lot of financial stake involved. So I'm a little discouraged about that. There is a bit of a success story in that originally and understandably, people, when they talked about deep sea mining, they just talked about the effect on the seafloor, which is really disastrous. There's, they like kind of rototill the seafloor, and people have come back like 40 years later and looked, and it's like, wow, the tracks are still there. It's unchanged, like nothing's recovered from it. So in that deep ocean, a lot of the processes happen over thousands or even millions of years. The nodules that they're harvesting, these metal nodules that are on the seafloor, they have accumulated. Like if you grow rock crystals or salt crystals, in your kitchen, like the salt coming out of the water and just growing gradually. That's how these nodules are formed in the ocean. And it can take like 2 million years for one to grow to the size of a tennis ball. So it's not a recoverable thing,
Right.
not to mention the organisms that are dependent on that. So originally the discussion about deep sea mining just really focused on these effects on the seafloor. But we did a workshop with a bunch of people led by Jeff Drazen at the University of Hawaii. And we basically showed that that's just the start of the process, is lawn mowing the deep sea floor. They have to take the materials up onto the ship, process them, and then they discharge the silt and the sediment that they don't want back into the ocean. And they don't send it back down to the seafloor. They just dump
Just
it
on the
below the
surface?
surface. I mean, they put it below the surface, so it's kind of like out of sight. It's not just pluming out
Perfect.
right at the surface. We'll just stick it down far enough so you can't see it. But it's like they're talking about right at 1,000 meters. And then now that we've kind of raised a fuss, they're like, oh, maybe 1,500 meters or whatever. But it's kind of, in the noise as far as if that's going to solve the problem. And the effect of these plumes, so the plumes are not going to settle out. They just are spreading out. And it's continuous, like 30-year lease, continuous discharge of just tanker truck after tanker truck worth of sediment into the water. And so that's obviously going to like really, really strongly affect the midwater ecosystem.
Yeah.
But so as a result of, I think, of this paper, largely, now that is part of the discussion of the effects of mining. Like you almost always hear when somebody mentions environmental impact of deep sea mining, they include a mention of the midwater. And I think it's important, not just because that's the habitat that I like, but if you are living in a small South Pacific island and your president is telling you how great it's going to be to do deep sea mining off your shores, you think it's not going to affect you, but then you have this sediment plume that's going to come in and it's going to cross the boundaries. It's not going to stay in its little box that they're supposed to mine in.
Yeah.
It's going to come wash up on your coral reefs and affect your island
How does it affect ecosystems in the midwater region?
in a lot of ways. So a lot of things in the midwater, which you can, which we can see from the sub, they eat particles. So it's called marine snow that's coming down from the surface. And ideally this is organic particles. So it's recently dead animals, or it's phytoplankton. It's like the plants that have grown at the surface, then they accumulate and they start to sink out of the surface waters, and that becomes a food resource for things that live down below. So a lot of things eat marine snow. Now all of a sudden your marine snow is not nutritious phytoplankton. It's like clay and silt.
Dirt, yeah.
Also there's a lot of other like heavy metals and toxicants in the sediment that are stirred up from the bottom. And so now you're injecting this, these metals that we're not supposed to be eating into the food food web. And then also so many things use bioluminescence as a way to communicate down there. So for finding mates, for finding food. And now they're going to be living, you just imagine like a smoggy day or something, London at the worst of the acid fog. Their sphere of operation is just going to be shrunk. Normally in the deep sea, things can see bioluminescence. It's really clear. They could see bioluminescence from 50 or 100 meters away, like the length of a football field away, they could see a flash of light. But now they're going to be living in this fog of sediment. So there's really a lot of potential impacts.
Yeah, I feel like I often used to think about the deep sea as like this murky area, but you're saying that it's like crystal clear.
It's very clear, yeah.
Other threats to the deep sea are there? There's deep sea mining, which as we know is a problem, gonna be a problem. What else? It seems like such an untouched place.
Fishing.
How does fishing impact?
Believe it or not.
Sea?
So like orange roughy was popular fish a while ago. I guess you can still buy it, but I think people realized, wow, these fish do not mature and reproduce until they're like 100 years old or something. So when you catch a food-sized fish, it's like a great grandpa type, situation. And when you fish that out, it doesn't recover very quickly because they're not reproducing on like a 3-year timeline. So as people fish down deeper and deeper, they were having a really disproportionate effect on the communities. Not to mention, if the net touches these seamounts, it's destroying these 1,000-year-old corals, which again are not going to recover.
You can fish that deep down?
Yeah.
Using what?
Just like a really long cable and a net with
like
a chain dragging across it. Yeah, they trawl across seamounts. There's also pollution. So people want to have like quick fixes for environmental issues. And usually those are not removing the source of the pollution. They're just
Band-Aids.
Putting the pollution somewhere else.
Yeah.
And so it's not really solving the problem.
Like the DDT.
Yeah.
Yeah.
Exactly. But so there's plastics. Of course, they can even get down into the deep sea. As a remediation method, people are saying, well, we can just put the liquid CO2 down into the deep ocean, inject it into the deep sea, and then it will just like kind of skip this step of having to be absorbed by the ocean, by the atmosphere, acidifying the water, and eventually getting down there.
Right, because it's a known thing that the ocean's such a big carbon sink.
Right.
Yeah.
And so they're just like, we'll just short circuit that and just pump the CO2 straight to the deep sea floor. And so obviously if you're an animal living on the sea floor and you see one of these pipes coming towards you, it's pretty bad news for them.
Yeah.
But yeah, I mean, I think even locally, things like, needles, cigarette butts, like all these things.
They make their way down, all the way down there?
Well, I'm just saying like our local ocean is also affected. But yeah, we do see weird trash all the way down at, a few thousand meters.
Like what?
There's been like shopping bags, like plastic shopping bags that you
All
could
the way
read.
down there.
Yeah. We saw a Sidewinder missile one time, like just sticking straight into the bottom of the ocean. I think it was a test missile, but it didn't, we couldn't tell. But we did find a mammoth tusk down there, which is another story. And fortunately, my lab tech used to work on whale falls. So she had a permit for mammal collections because otherwise we wouldn't have technically been able to collect this mammoth tusk, even though it was like 160 miles offshore. But
where is it now?
It's in Michigan. It's at the museum, the paleontology department in Michigan.
At University of Michigan?
Yeah.
I've seen it. I went to school there. Yeah, I've actually seen it. I didn't know that you guys collected it. That's wild. Okay. So what are you hoping through your research, MBARI as an organization, and this network of marine biologists around the world? Like, I guess, what are you hoping the future looks like for the deep sea and maybe even specifically Monterey Bay?
I mean, I think at the highest level, the goal is to increase the awareness and the investment, like people's personal investment in the deep sea. There are more specific things like, well, let's quantify the amount of microplastics in the ocean, or let's look at trends in oxygen, like oxygen levels through the years or decades, and see, how that's changing. But I think the awareness part of it is really the main thing. There's also, I think we can have some impact in the genetics part of it. Like people want to have the easy part, which is I don't even know what the name of it is because I didn't watch Star Trek that much, but they have like some little thing that they point at an organism and it tells you if it's an alien or a human or something.
Okay.
Anyway, there's like, this dream of like handheld device. It's like going to just collect DNA from the environment and analyze and see what's in there.
Yeah.
But in order to get to that point, we have to have the database, the reference database of like, here's all the animals and here's their genetic sequences. And here's, we actually know that these are the same species or whatever. So we're trying to do that part of it, which I think will open the door for these future, more automated applications. So that's why we're collecting animals and sequencing their genes and documenting them, and just trying to assess the full measure of diversity down there.
I think that's what really does set the Monterey Bay Aquarium and MBARI apart is the amount of work they do online to push out, like, this is what we're seeing, video footage of you guys on the ship or what you guys are seeing from the ROVs. It's pretty incredible. And to my understanding, you have a science communication team too, which does some awesome work.
Yeah.
I'm wondering, is there any way that the community can get involved through MBARI or maybe citizen science initiatives you guys have?
Yeah, I was thinking citizen science. I mean, there are opportunities to volunteer at the aquarium, but we don't really have anything like that at MBARI.
Mm-hmm.
The easiest way that people can translate their experience at the beach into like actual scientific data is through something like iNaturalist. So you're walking on the beach, you see a jellyfish wash up, take a picture and submit it. And that actually comes in handy later when we're trying to say, well, when did the bioluminescent sailors wash up on shore last year and the year before and the year before? Is it getting earlier, later? What's their distribution along the coast? Those kinds of citizen science observations cumulatively end up being really, really valuable. I do realize that I'm in like a very privileged position where I am, the resources and the access that I have. And so I think part of my responsibility is to try to share what I see with other people and, both through our publications in science, but also making YouTube videos or, articles in popular press and media. I think, like you said, the more that we can have this communications team reach out with people, and I think then it's part of our responsibility, really.
All right, so I figured we'd end our conversation with a lightning round of questions.
Okay.
So I'll ask you a question and then give me first thing that comes in your head, quick response. Here we go. First question is, if you weren't a deep sea scientist, what would your alternate career be?
Like realistically or fantasyly?
What, you can do both.
I mean, I think if I was more talented, I think my alternate career would be like a graphic designer or something like that.
Okay.
I think that'd be really fun. Or be in a band, I think would be really fun.
Do you play instruments right now?
I mean, I play cello and a little bit of guitar and bass.
Well, you could still be a band on the be in the
I
band
know,
on
I
the
know,
side.
I know.
Like, all right. Question 2: what is the best piece of advice you've ever received?
I had a professor, Alice Aldridge, who I would go on blue water diving cruises with, and we would be like not in the tropics, but in pretty cold water. So you're like getting into a cold wetsuit multiple times a day. And near the end of one of those cruises, I was pretty tired and was deciding if I should go on this night dive. And she's like, you really should take advantage of like when you're in the field, those opportunities are irreplaceable and you can always like rest when you're back home on shore. So she's like, we should go diving. And then my aunt, who got her PhD in rhetoric, but she, when I was kind of struggling, like I have these perfectionist tendencies.
Me too.
And so she said the best thing anyone ever said about a PhD is that it's done. Don't try to make it perfect, just try to make it done. So that was,
Don't try to make it perfect, try to make it done.
I often think about that.
All right. Question 3: what's your favorite outdoor spot in Santa Cruz or in general, the Monterey Bay area, to just get like your quick fix of nature? And it can't be the deep sea.
I mean, I guess south to north, Point Lobos. I love going to Point Lobos, and we have a secret beach near Point Lobos that I don't want to tell you about, but I'm sure you can figure it out. And then around Santa Cruz, like Mitchell's Cove, or body surfing at Its Beach, 4 Mile. The Pigeon Point Lighthouse, there's a tiny beach like just before the parking lot. If you go down the stairs, there's like a little hidden cove there that I just love. It's tiny, but it's really amazing.
I'll have to check it out. Okay, number 4. You can give me a book, a movie, a show, or all 3 that you would recommend to a science lover.
The book I read recently that I liked was An Immense World by Ed Young. He actually came out on the ship with us a little while ago, for his next book. But his book, it looks at the different ways that animals perceive their environment besides just like our normal familiar senses. And it's a really good book.
Can you detail what his next book is about? You said it was no. Okay.
For the movie, there's a video called depending on if you're in England or in US, I think it had different names, but Life That Glows or Light on Earth. It's like a David Attenborough thing. But it has some really cool, one-of-a-kind bioluminescence footage in there. And then I'd have to plug our book, I think, because
I was gonna say, are you?
I gotta mention it just because, like, that is where I put like what I think about the ocean or how I see the ocean.
Yeah.
So
and I flipped through it. The photos are incredible. So
thank you.
Yeah.
Yeah. So that was definitely a labor of love, and I feel like that gives a different perspective on the ocean. So. Definitely.
Yeah. We'll link that in the show notes. Yeah. Okay. Last question. What gives you the most environmental hope right now?
Again, it's you got to cling to every little opportunity that you have. I think one of the things is like the students that I interact with. So I gave a little talk for like a kindergarten class with these little kids, and they're like, I saw crab on the beach and stuff like that. And they're just like really into it and super enthusiastic. But even up to like grad school level students who are just super passionate about, saving the world. And so probably interactions with students.
I think it's important to not let the doom and gloom touch them just yet. Like,
I know.
Let them have their youthful optimism, because I think we all need that. So yeah, I think that's important. All right. Well, thank you so much for having this conversation with me and taking us all out to see the bottom of the ocean today. It was a pleasure to have you here.
Thanks a lot, Cassidy.
All right, that does it for the pod. But hey, before we go, would really love your feedback about these Field Diary episodes and just your feedback in general about the program so far. So send me an email. It's jahicken@ucsc.edu. Of course, you can also leave comments on YouTube and on socials. I read all that stuff, so would really love to hear what you like, what you don't, so we can continue to make this program better. But for now, thank you to Steve Haddock for coming into the studio and for taking Cassidy and the team out onto the vessel with you. Thanks to everybody at MBARI for your incredible work. Thank you to Cassidy, of course, for guest hosting and collecting all that footage at sea. Thank you to Edith Belmont for editing, Valerie Situ for production, distribution, and socials. Thank you for listening. We'll see you next time.